So I'll start off by reviewing metabolism under normal aerobic conditions. This is a gross oversimplification, so if you're not familiar with glycolysis, definitely there's lots of videos out there on it. Go and learn that. So in this process, glucose is converted into two pyruvate molecules, and it requires two NAD plus and two ADP. And it gives us two NADH plus 2 protons and 2 ATP. So that ATP can be used as energy right away. The NADH is going to be converted into ATP later by the electron transport chain. Again, this is a gross oversimplification, but I'm going to quickly walk through aerobic respiration. So that pyruvate that we made in glycolysis is going to enter the Krebs cycle as acetyl-CoA. And here it's going to produce CO2 and more NADH. This NADH can enter the electron transport chain, where the electrons from NADH will reduce oxygen to H2O, which will allow us to produce ATP and regenerate that NAD plus that we used during glycolysis. Now this is what happens under normal aerobic conditions, but in the absence of oxygen, like if you're exercising strenuously, NADH cannot enter the electron transport chain, so we can't reduce oxygen to H2O, we can't produce that ATP, and we can't regenerate NAD+, and eventually the Krebs cycle will shut down as well. Without that NAD+ coming from the electron transport chain, we're eventually going to run out and glycolysis is going to come to a halt. So we can't produce those two ATP. So we're going to have to find a way to get that NAD+ back from pyruvate. This is where lactate fermentation comes in. And it's important to know this is not anaerobic respiration. Anaerobic respiration still uses the electron transport chain. It just uses something other than oxygen as the electron acceptor. So the general reaction of lactate fermentation is pyruvate is transferred into lactate. And in the process we consume one NADH, which was a product of glycolysis, and we make an NAD plus and a proton. So we're regenerating that NAD plus that we used during glycolysis. Now that we've reproduced that NAD plus, glycolysis can resume and we can make those two ATP from each glucose. This is called anaerobic glycolysis. The enzyme that catalyzes lactate fermentation is called lactate dehydrogenase, and it's actually a really important enzyme, so I'm going to take a closer look at how that works. It's made up of 4 subunits, and it has a reaction center in the middle. So I'm going to take a closer look at that reaction center. There are 4 amino acid residues that stabilize this reaction, and the reaction is sequential. So the binding of substrates, the order is important. NADH has to associate first. And then you can see it makes this area in the middle that's going to stabilize the binding of pyruvate. So the carboxylate group on pyruvate is stabilized by arginine 171 over here on the left. And then the keto group is stabilized by arginine 109 and histidine 195. You can also see that this histidine is stabilized by this aspartic acid residue, and we've seen this before in our reaction trios. So that's going to stabilize the histidine so that it can be a proton donor. Once pyruvate is in the active site, this carbon becomes partially positive. So now a hydrogen from NADH and its electrons can come up and attack this carbon. Then the double bonded electrons will move up onto this oxygen, which picks up a hydrogen from histidine. So now we've generated our final lactate product. Because this is a sequential reaction, lactate has to dissociate first. Then the NAD plus can dissociate, and finally this histidine 195 is reprotonated by the extra proton we created during glycolysis. If you've been paying close attention, You may have noticed that the name lactate dehydrogenase does not describe the reaction we just looked at. We just added 2 hydrogens to pyruvate. Now the name lactate dehydrogenase would suggest that we're removing hydrogens from lactate, and that's actually what the reverse reaction does. It's very important that this reaction is reversible because lactate is a metabolic dead end. It doesn't actually do anything. In the absence of oxygen, lactate concentration is high and pyruvate concentration is low. But once aerobic respiration resumes, pyruvate is converted back into lactate. And so pyruvate concentrations are high and lactate concentrations are low. And this follows Le Chatelier's principle. So during fermentation, lactate actually acts as a pyruvate reservoir. Before I wrap up, I want to cover some of the diseases and conditions that are associated with lactate dehydrogenase. Lactic acidosis is the buildup of lactate, and it happens during intense exercise, and it's also a side effect of some illnesses. During a myocardial infarction, oxygen-starved cells undergo involuntary lysis. So lactate dehydrogenase is elevated in the blood in the days after a heart attack. And you can actually test for lactate dehydrogenase in the blood to see if somebody has had a heart attack recently. In cancer, tumor cells often have increased lactate dehydrogenase expression, and this makes sense because one of our natural defenses against tumors is to restrict blood flow to that area. So they're going to want to do a lot of lactate fermentation so they can keep making ATP. And finally, lactate dehydrogenase is abundant in red blood cells. So seeing lactate dehydrogenase in the blood is a sign of hemolysis. Finally, I'm going to talk a little bit about alcohol fermentation. Now, luckily, we don't get drunk when we go for a run because we don't have the enzymes to produce alcohol in our muscle tissue. However, alcohol metabolism of alcohol that we drink does produce NADH, which is a substrate for lactate dehydrogenase. So this increases lactate production, and a secondary effect of severe alcohol poisoning is lactic acidosis. And then if you can imagine, if you do strenuous exercise when you're drunk, you're going to have lactic acidosis symptoms. So these guys here are about to do the beer mile, which might sound like a really good idea, but I actually don't recommend it. It'll make you really sick. So that's lactate fermentation. Thanks for watching.